Yeah. Thanks so much. Thanks, Holm. Thanks, Han. Thanks, everyone. So obviously, you'll understand I'm standing here on behalf of many, and in fact, almost on behalf of all of you. So thanks for the trust in us and for staying with us so confident to really push this towards the finish line. And you'll see there's a little bit of redundancy in there, and some of the pictures came by, but I just want to give you the coherent story of what we call indeed the flagship paper for work package 2 for the reanalysis part. So obviously it's almost needless here for this audience to explain that rare diseases are just so important, especially because they're collectively a very common health issue. And we set the standards quite high, and we discussed that extensively when we wrote Solvardi, that we will only invite to very actively participate centers where exome sequencing is already standard, at least for some of their cohorts. And we knew that is leaving some behind, but we wanted to start with that to really set the example here. And while exome sequencing is so widely successful and hugely successful, even there we know by now that the success rate in terms of diagnostic yield varies between 20 and 70%. And I think across Europe or across the world, we don't even see huge differences. So it does not matter so much how and where you do that. It's just intrinsic to our current knowledge, and even the best centers still succeed by reanalyzing the data over and over and over again. And I think we're demonstrating that once more in this exercise. So amongst the many goals of Solvardi, as Holm also alluded to, we said, okay, we need to get our act together and really bring together data and expertise throughout Europe. And that's in essence what we did here. And we did so with a core set of data that we termed RD-Real, which stands for Rare Disease, after Reanalysis Logistics. That's bringing together data sets, and we qualified the most important bits and pieces that we always need. And that is the genomic data, usually the exomes, or occasionally the genomes. That is ontology-based phenotype description of all cases, and essentially also the information about the pedigree. And what I'll present now is the first very systematic reanalysis of what we call data freeze 1 and 2. And that represents the data of 6,004 undiagnosed rare disease cases or their respective families. This is reiterating it. This part, data freeze 1 and 2, are using 4 of our core ERNs, our earliest partners, all 4 of them, being R&D, Ithaka, NMD, and Genturis. And if we count that up, that represents more than 300 collaborators and 37 institutes from all over 12 European countries, plus Canada. If you then look again at the data sets that we collected, it's close to 10,000 genomic data sets after QC. On average, for all individuals, we have 6 HPO terms, but obviously there is a wide spread. And we had the detailed phenotype pedigree information for all families slash cases. We then set up various instances in Solvardi. Those are what Holm also showed on the previous slide. For this specific reanalysis part, 5 working groups within the DATF. For each and every ERN, a specific dedicated DITF, Data Interpretation Task Force. And this is really the ping pong between the more molecular and bioinformatic experts and the real disease experts in the DITF. And I think it's this ping pong game that we played for all so long that really, first of all, educated ourselves, but it also proved to be hugely successful. And I think here we found this very pragmatic solution that usually on a global scale may take years. So people sometimes upload their data to databases, to a national database, then discuss, and then it's sometimes fed back. But that process usually takes years until you make from a variant of unknown significance, finally a proper diagnosis. And with this direct ping pong between these 2 levels of expertise, we really succeed now much faster. While it still sometimes is feeling painful and still feels as if it takes long, in reality, if you compare that on a global scale, it really is efficient. And just to illustrate that, Holm mentioned that briefly as well, we calculated how much time we spent on this 2-level expert review of data to make final calls on a set of variants per case. And for many variants, the experts all said, oh yeah, this is clearly benign, clearly has nothing to do with the phenotype. And then for a significant proportion, they took all the consensus decision that it was so important and relevant for disease. So with all that in place and established over the last years, we moved forward, and I'll just try to very high level summarize the reanalysis yield. So here you see again, we start with 6,004 families after QC. We offer our comprehensive analysis suite, and then we have the ATF working group expert level, and then this ping pong between DITF disease experts happens. And then we come to the following consensus and following finding. We make a new genetic diagnosis based on point mutations and indels in 419 out of the 6,004 families. In an additional 87 of families, we identified a novel, some may see even more exotic variant type that goes beyond point mutations and also goes beyond what most centers, even in the diagnostic setting, use as a standard. Additionally, and very importantly, we're also fully sharing additional candidate genetic variants for 333 families where we have quite a high evidence that they're likely involved in disease, but not the final proof. And I think this we really have to share with the global community because I sincerely believe that of those 333, there may be other cases globally that have the same variant or even the same phenotype, or ideally both. And then we not only solve our case, but we also solve and diagnose a patient elsewhere on the planet. So that's really our global duty. I think we also fostered all the individual centers by systematically getting all their data together to also run more systematic analysis in-house. And you can term that an ad hoc expert review that happens usually locally. Even that was largely successful, and some of that obviously happened in parallel or in the background. And that solved, and sorry for the terming, it's just the solver phrasing. Solve, I think, is a terrible term. It diagnosed an additional 249 families of the 6,004. And now on the next few slides, I'll go into the details of the yield that we had on this slide. This is just summarizing that still, even after this exercise, there's 82% of cases where we cannot provide a better diagnosis at this stage. And this pie chart is just a different visualization of what I've shown you on the other slide. This may be more interesting, maybe. This is splitting the respective categories of yield across the 4 core ERNs. And you see that there is differences. But if you would normalize for the number of known genes for the respective disorder, it's actually quite similar. So this is largely a reflection of the well-established disease genes for the respective ERN. So if you would now naively assume that all these diseases are similarly heterogeneous in their genetics, then I think we could just normalize for that. In a few years, we may be there. And it may, to some extent, if you just give the most extreme example here, if you would assume that a lot of the familial cancer syndromes are in fact genetics, but we're just lagging the current knowledge about a majority of genes, I think there's even more potential to find more new diagnoses in that category particularly. So we could even see that as an opportunity. Just to give the absolute numbers again as a reminder, this is the numbers of families that now finally can receive a diagnosis after the systematic reanalysis, 506, the local ad hoc review, 249, and additional candidates that are still like in this limbo and hopefully soon will get a proper diagnosis for 333 families. Again, if you're interested in more detailed numbers, that's all obviously in the supplement, but I know sometimes people are really keen on seeing the exact percentages per EN and disease category, but I won't go into all details of this specific table for now. Now, yet another level of zoom in. And we did that particularly for the point mutations because it's the largest group. Here we asked the very simple but important question, why didn't get the respective cases and families and referring clinicians a diagnosis earlier? What is the issue here? The answer is, in 165 of the 419 families, it is simply a re-annotation of variants. So since the original submission, our knowledge on variants that properly cause disease has increased, largely by knowledge collected in ClinVar. And in fact, this wouldn't need solver-D. I think a lot of local centers could do the same trick. It's just updating your existing data locally with a great knowledge of ClinVar. So, and I think it could be automated to an even more massive extent, and it could be like almost done live in some instances. The second category of why a diagnosis wasn't found originally is that the gene wasn't published at the time of submission as a disease gene with disease-causing mutations in it. And that held true for 63 of the families. And then a large chunk of cases were only solved by our two-level expert consensus. So here, I think Solfady really played the most important role. It's really the different levels of expertise we assembled all over Europe and really had the experts at the table that collectively came to a consensus saying, yes, this is not only a pathogenic variant, but it also fully explains the phenotype. And that is a large chunk, and all of those data are obviously now also uploaded to ClinVar. Should I emphasize for these 191? We're probably the first finding these variants. So they're largely private variants and haven't been seen, or at least are not in the public domain. So importantly, we feed that back to the public domain to enable others to benefit from this expert judgment. If I summarize per inheritance type, that's the little table in the upper left here. Upper right, sorry. If I again then zoom in into the variant types, as I mentioned, large proportion of the diagnosed cases is based on S and V indels. But then in additional data analysis working groups, we also looked at other more exotic variant types. As already emphasized by some of the previous talks, there's even related manuscripts either in revision or submitted as part of the bundle. And I'll just go into those in detail. If we count up these non-point mutation and indel variant types that are not standard, that explains a total of 87 families, as mentioned in one of the previous overviews. Here are two types that are actually point mutations, but are not necessarily seen in each and every standardized pipeline. Those are non-canonical splice site variants, as you can see on the left, or mitochondrial DNA variants that may in some centers be studied, but not in all systematically enough. And again, for those, we found quite a significant amount of samples with the level of expertise Solfady provided. Then moving to the larger variant types. So those are CNVs, as you can see on the left, or those are even very exotic events where exome sequencing really isn't suited at all. But if you do it in a large enough cohort, it still explains individual cases and gives a diagnosis to individual families, so still valuable. And then there's additional ones, again, similar to the mobile element insertion structure variants. They're certainly, or exomes are not very well suited to find those. But still, if you try those tools on such a large data set, there's individual ones where they really matter. And I think, by the top of my head, it's 7 SVs in our cohort in the reanalysis. Similarly, short tandem repeat expansions are, again, not the primary variant type you can identify in exomes. It was never designed in such a way. But again, if you apply it here systematically with all the expertise on board, we make new diagnoses based on those. I'll just also bring up 2 examples to make it even more tangible of what we really do here. So here we had one case from the Ithaca ERN, and the diagnosis was finally made based on a mosaic variant with a variant allele fraction of just 13% in a by now very well understood gene. But the standard filter settings in the respective center were, I think, 20%. So the standard output in a diagnostic setup does not identify this variant. The original medical diagnosis you can see on the left-hand side for these 24-year-old male patient. Originally, the clinician remarked that it's most likely an underdevelopment of the left side of the body. By doing our systematic reassessment, the very same variant was already in ClinVar by now, and we didn't apply initially a variant allele fraction filter, but just highlighted this as a variant that is present as pathogenic in ClinVar. And then you can see that it's 13%, and by looking at the other presented phenotypes in ClinVar and publications, the diagnosis actually is changed. It's not an underdevelopment of the left side, but rather an overgrowth syndrome of the right side of the body. So even there is something important for the clinical lecture of this case. Second example is from our partners in R&D. So for the rheological diseases. And this is one specific patient that had a progressive gait disturbance and a few other features, including spasticity, ataxia, and so on. And there was already a point mutation identified originally in the exome sequencing data set, but the second allele in this somewhat well-matching gene was still hidden. And by the systematic reanalysis, we identified a 10 kb deletion that affected exons 6 to 11. And with this at hand, additional tests were done, including plasma glycolipid tests that really provide functional evidence that indeed we really have the diagnosis here and the diagnostically disrupted gene. And with that, a final diagnosis could be made for an autosomal recessive spastic paraplegia of type 26. All of that said, we can obviously even move a little bit beyond diagnosis. And this is by just asking the very simple question, of the 506 families that finally received the diagnosis after our reanalysis, how many of those would be amenable to some kind of actionability? And that is close to 15% if you count that up. However, the trouble there is, this is really bringing together the knowledge that we again have jointly based on the tree tabalome, but some other publicly available data sets. But there you see the overlap is very limited. So it means that this is still largely fragmented in its nature. So I think we can get much better as a community, and we invite everybody to get engaged because now we can use that more systematically. And I think we all envision that the diagnostic report is not ending on the molecular diagnosis, but we should give every referring clinician at hand saying, hey, are you aware that this is potentially actionable? So I'll just sum up by the lessons we learned, or some of the lessons we learned. I think it's good and important to emphasize that even in these proper expert centers all across Europe that are all individually extremely good, we are far from perfect. So our knowledge about genetics and even monogenic diseases is not saturated at all, even for these very well studied ones. And that means we still learn new information each and every week on gene level. Novel disease genes are still discovered, and also importantly on variant level. And only by such a reanalysis we can do that. And I think we really have to emphasize that we all need to do that either very regularly or ideally at some stage live. It's also important to emphasize that while exomes are never perfect for other variant types than point mutations and indels, there is a significant amount of data that you can still extract from it by applying a more systematic reanalysis suite of tools. And in our case, that diagnosed close to 16% of cases. One final statement, numbers really matter. And they matter on two levels. I think this very large collection matters because we can really feed into the global network, for example, ClinVar or other matchmaking resources. And while all our cases are really super rare, there may be a second case in the US or in Australia or somewhere in Africa. So it's, I think, our global need that we really need to bring this and the real numbers together. But numbers even matter within our cohort. Even within the 6,004 families, there were 21 variants that were identical in 2 or 3 cases. And obviously, bringing those two experts in Europe together that may have never met each other is extremely useful to make a more firm diagnosis, and it helps dramatically. I mean, we can do a lot of functional work, and I wouldn't argue against that at all, but we are so much better in phenotyping humans than we're in phenotyping mice and other experimental models. So I think really these numbers matter, and we really have to continue to engage here. As highlighted, close to 15% are potentially actionable or even treatable. And as Holm already emphasized, I think this may actually be a blueprint for similar endeavors on a global scale. I think in particular, the framework we present says framework, actually, nice typo. So this framework seems to work. And in particular, the ping pong between different levels of expertise on a European scale, I think really is what pushed this forward. And as Holm said, by now we know that on average, if you look at the most common variant types, we find on average 9 to 10 variants per case. If we calculate how much ping pong between the experts happened for this, it's 42 minutes per case. So for everyone from the EU, we can now calculate how important and how difficult, how much this would be for all rare disease cases across Europe and how much that would cost. And I think it's all feasible. With that, ending just with a very important note, this entire resource is open to the entire community. So as you know, everything is deposited under controlled access in line with all the consent everyone here in the room actually took care of. And it's available in multiple formats. It's available in raw formats for people that really want to do massive reanalysis, but it's also accessible in a very interpretable form for individual clinicians and clinical scientists that can look up now variants in GPAP, whether that was found before or whether a similar phenotype was found before. So there's various levels how we make the data available. And I think that's what we had to do and what we, I think, delivered. With that, as I said, it's really talking on behalf of many, many of you. Again, thanks for all your efforts spent over the last 5 years to make this happen. There's several people highlighted here. We have 6 shared first authors, 6 shared last authors. and many, many others that are really put in name here in all the efforts for this specific paper. But in fact, this is just representative for the 4 core ERNs and even the entire consortium, obviously. So I'll leave it at that, and thank you very much for your attention.